Alright, we will continue to look at how transcription is terminated once the gene sequence has been transcribed and once the RNA polymerase moves to the terminated sequence. Alright, so once it reaches the terminated sequence, the RNA transcript, the mRNA, right, would be released, and of course the double helix of the DNA would rewind, and the RNA polymerase would leave and ready to bind to another promoter if there's a need to transcribe another gene, right? Or if it needs to transcribe the same gene again, then we just go back to bind to the same promoter. Okay, so for the prokaryote, right, once transcription is finished, this mRNA is immediately functional. It will go on to serve as the template for translation. But for a eukaryote, right, this RNA, this mRNA, right, is known as a pre-mRNA, which suggests that it's not the final mRNA yet, right? It's the pre-mRNA. So this pre-mRNA needs to be modified in a process of post-transcriptional modification to form the mature mRNA. So the mature mRNA would then serve as the template for translation. Right, so the post-transcriptional modification, we will look at it next lesson. Okay, so for now, we'll look at how transcription is terminated first. The process is different, right, between the pro and eukaryotes, so we look at them one by one. For the prokaryote, the terminator sequence, right, contains a sequence that is self-complementary. All right, so this is the DNA sequence, right? And within the terminator sequence, there's this part that is self-complementary. So once the RNA polymerase has transcribed the terminator sequence, the mRNA also contains a self-complementary region. Self-complementary, so for example, if this part is say CCGGA, right, then this part would be complementary to CCGGA, which would be UCCGG, right? So because the terminator sequence contains a self-complementary part, so after transcription, the mRNA also contains a self-complementary part, right? Because the mRNA is complementary to this. So if they are self-complementary, the mRNA here and here will also be self-complementary. Okay, imagine that, right? So when that happens, right, because they are self-complementary, then the mRNA can fold into a hairpin loop structure. And this hairpin loop structure would somehow block the movement of the RNA polymerase, and that cause the RNA polymerase to stop moving, and then it will then dissociate from the DNA, and that marks the end of transcription. Right, so again, I have some animation to make it clearer to you. So this is in the DNA, this as part of the terminator sequence. As you can see, right, they're self-complementary, this part, and this part are self-complementary. Okay, so once transcribed into the mRNA, so remember the terminator sequence is transcribed, right? So this would be the corresponding complementary sequence in the mRNA corresponding to the terminator sequence, right? And they are self-complementary, as you can see, right? So it allows for, so you can imagine the mRNA now kind of folds. So the C can bind to G, this C can bind to G, right? This G will bind to the C, and so on. Right? So it forms the hairpin loop, which dissociates the RNA polymerase, and therefore stopping further progress of transcription, also that terminates transcription. All right, for the eukaryote, there's no hairpin loop formation. It's a bit different. The terminator sequence, right, carries a particular sequence with this AAU, AAA. Okay, so again, let me just repeat this to make it clearer. So the terminator sequence is transcribed, right? Within the terminator sequence, there's a particular sequence in the template strand that codes for a polyadenylation signal, right? So, the DNA sequence, right, in the terminator sequence, Okay, this is a mouthful. So within the terminated sequence, that would be this part with a sequence of TTA, TTT, okay, in the template strand. Okay, so the RNA polymerase transcribes the terminated sequence, right? So in the mRNA, there will be this AAUAA sequence corresponding to where the terminated sequence is. Okay, so this AAUAA sequence is known as a polyadenylation signal. Right, so some other proteins would recognize the presence of this AAUAA sequence in the mRNA, and they will come to cut the mRNA. Okay, so once the mRNA is cut, of course, then the pre-mRNA would be free to leave. Then there will be other mechanisms that we actually do not really understand very fully to stop the RNA polymerase, and the RNA polymerase would go on to dissociate from the DNA. Okay, so some suggest that those proteins that cleave the mRNA also go on to dissociate the RNA polymerase, but we don't know this for sure. This part is still, from as far as I can find online, right? This part is still under investigation. How the RNA polymerase leave the DNA, right, in the eukaryote. Okay, but in terms of the mRNA, right, it stops elongation because it is cut away, right? It's cut, so the pre-mRNA leaves. All right, due to the presence of this AAUAA polyadenylation signal in the mRNA. Okay, and this polyadenylation signal comes about because of a corresponding complementary sequence found in the terminator sequence. Okay, all right. Okay, so this pre-mRNA, right, as we mentioned earlier, right, goes on to undergo post-transcriptional modifications, which we will look at in the next lesson. Okay, so as a summary of the whole process of transcription, right, the RNA polymerase binds to the promoter sequence somehow, right? So it's the mechanism of binding is different for the pro and eukaryote. We call the signal factor versus the sigma factor versus the general transcription factors. But once the RNA polymerase has bound, right, there will be the unwinding, then transcription will begin, and the mRNA is elongated 5' to 3'. And once the RNA polymerase reaches the terminated sequence, right, then transcription will stop. Right? And how the transcription stops is with different mechanism again between the pro and eukaryote. We call the hairpin loop versus the polyadenylation signal. Okay, so this would be a summary of transcription process. Okay. So a question here, right? Why must the DNA be first copied into mRNA? Right, this is the question, right? Why have this extra step, right? Why can't we straight away use the DNA to make the protein in translation? Why is there a need for the DNA to be copied into mRNA, which basically carries the same information, right? So why copy something that is rather the same, right, carrying the same information? Why don't we use the DNA straight away for translation? Now think about it. What's the advantage of copying into mRNA? Okay. Okay, so some possible reasons through evolution would be, right, we don't want the DNA to be exposed to so many things, right? For a eukaryote especially, the DNA should stay in the nucleus for protection, right, to prevent damage, prevent mutations. Okay, so, but for a eukaryote especially, right, the translation only occurs in the cytoplasm, right? So whatever template that serves as a template for translation needs to leave the nucleus and enter the cytoplasm where the ribosomes are, right? So we don't want the DNA to do that. We want the DNA to stay in the nucleus, right? So that's why we need the mRNA to carry the same information from the DNA out to the cytoplasm, right, for translation. So then the DNA can then stay in the nucleus and remain protected. And also, right, there's only like one copy of a gene in a cell, right? So if we use the DNA as the template, then there's only one template for translation. But if we can carry out transcription multiple times, then from the same gene, we can keep transcribing the same gene again and again and again. So one gene can produce like many copies of mRNAs after multiple rounds of transcription of the same gene. So we have now more templates for translation, so more proteins can be made faster. Right, so this is not possible if there's no mRNA and we only use DNA, and then there's only one gene for each one copy of each gene. There's only one template for translation. Then the protein synthesis will be quite slow. So two possible advantages to having mRNA, or if you think about it, right? Okay. So that would mark the end of transcription.